In this video, I'll be going over a few of the common mechanisms of antibiotic action. These include inhibition of cell wall synthesis, inhibition of protein synthesis, and alteration of membrane permeability. One of the most common mechanisms of antibiotics is the inhibition of cell wall synthesis. This is because human cells do not contain cell walls. So when an antibiotic is administered to a patient, you do not have to worry about it interacting with the host cells. One of the main functions of the bacterial cell wall is to protect against differences in osmotic pressure. The gram-positive cell wall, in this example, is made up of a thick peptidoglycan which contains NAG and NAM subunits, which are cross-linked for structure and stability. Penicillin is an antibiotic that works by blocking the action of the enzyme transpeptidase, which cross-links the NAG and the NAM subunits. Vancomycin is an antibiotic that takes it a step further and blocks the subunits of NAG and NAM from interacting with transpeptidase. These antibiotics lead to a less stable and weakened peptidoglycan structure, which leaves the cell vulnerable to lysis and death in different osmotic conditions. Another mechanism of antibiotics is the alteration of cell membrane permeability. Two examples of antibiotics that do this are polymyxin and bacitracin. Cell membranes are one of the most important parts of a cell, whether it's eukaryotic, prokaryotic, gram-negative, or positive. In gram-positive bacteria, it contains just one cell membrane inside of the cell wall, while in gram-negative bacteria, it contains both an outer membrane outside of the cell wall and a cell membrane inside the cell wall. The membrane is selectively permeable, so it is what controls what enters and what exits the cell, whether it be good or bad. Throughout the membrane, there are proteins which function as channels. Here. These only let certain things in because most cells need a specific concentration compared to the outside environment. If you alter the membrane, it can have dire consequences. Cells usually contain higher concentration of things like macroelements and growth factors within the cell, here, along with a higher concentration of water outside the cell. If membranes are altered to be more permeable, it can lead to a loss of the macroelements leaving the cell, along with a great influx of water into the cell, which could eventually lead to lysis. If a cell relies on proton gradient and proton motive force, it could be rendered useless with the inability to contain ions. Since somewhat similar membranes are also present in eukaryotic cells of the human body, these types of antibiotics are rarely used internally, and their use is typically confined to topical applications. The last mechanism of antibiotics I will be talking about is the inhibition of protein synthesis. Like the antibiotics of cell wall inhibition, these can be used safely in humans because they affect the 30S and 50S ribosomal subunits that are not present in eukaryotic cells. Inhibition of protein synthesis does not necessarily kill bacteria right away, but it does prevent them from multiplying and growing. For this reason, they are said to be bacteriostatic as opposed to bactericidal. Certain antibiotics only affect the 30S subunit. Examples of these include aminoglycosides and tetracyclines. Aminoglycosides bind to the 30S subunit, affecting its formation, while tetracyclines block the tRNA binding to the subunit. Clindamycin and macrolides are examples of antibiotics that affect the 50S subunit. Clindamycin affects the peptidyl transferase, which catalyzes the peptide bond formation between amino acids during elongation, while macrolides act by binding to the 50S subunit, preventing peptide elongation. The two other common mechanisms of antibiotics that I did not talk about was inhibition of nucleic acid synthesis and altered enzyme function. And in conclusion, if there's one thing that you should learn from Dr. Cooper's microbiology class, is antibiotics have no effect on viruses. Because if you say it, he will make fun of you for it.